Welcome back for this afternoon session. It will be on study designs. I'm Dr. Savarese from the Karolinska Institute in Stockholm. And after me, Dr. Nissen talk about randomized clinical trial. So we start with observational studies and registries. So I know I don't look like a Swedish guy, but that's a long story. So actually we have two different types of studies. This is the first classification. We have descriptive studies that just describe frequency, natural history, and determinants of a condition. And we have analytic studies describing association between exposure and outcome. We can divide studies in two groups: experimental studies that can be randomized or not, and observational studies in which we can have a comparison. These are the analytic studies, and they can be cohort study, case control study, or cross-sectional study, or if there is no comparison group, just the description of a disease, we can talk about descriptive study. Different type of studies, different quality of evidence. As soon as we go from an expert opinion, case report, prospective cohort, controlled clinical study, to randomized control trials, to systematic review, we increase the level of the evidence. Observational studies can be divided, are not interventional, are not controlled, and they can have a prospective or retrospective design. These are the main characteristics of observational studies, so let's see what this means. So in a non-interventional study, the investigational medical product is already authorized. The investigational medical product is used in accordance with the terms of the marketing authorization of the member state concerned. The assignment of subject to a particular therapeutic strategy is not decided in advance and doesn't fall within normal clinical practice of the member state concerned. The decision to prescribe the investigational medical products is not taken together with the decision to include the subject in clinical study. Diagnostic or monitoring procedures, in addition to normal clinical practice, are not applied to the subjects. These are the rules of not interventional studies according to the EU regulation. So we say that observational study can be prospective or retrospective. What does prospective means? Prospective means that if the study starts today, and today we assess the baseline characteristic of the patients, we will look for clinical events in the future. Retrospective study means that if today we consider the baseline characteristics of patients, we investigate if in the past there has been an outcome or clinical events. Then we also have cross-sectional designs. In cross-sectional designs, we consider the population characteristics regardless of time. So it's like the study exists only today, and today we assess the relationship between the exposure and the outcome, but we will see this in particular. So, as I was saying, for cross-sectional study, we don't consider the follow-up, so it's just now the point in which we want to consider that the study exists. So we consider the study population, we discriminate between patients who have the outcome or who don't have the outcome, and then we assess if the exposure or factor was present or not, and we compare the study groups. The cross-sectional studies are the simplest one, have merits and limitations, obviously. The merits are that they are quick, they are cheap, they allow to study several diseases and exposure at the same time. They are very good to assess the prevalence of a disease, and they are very good for public health planning. The limitation is the temporal ambiguity, because as I said, the study exists just in one time point, regardless of follow-up, so regardless of what can happen tomorrow or what happened yesterday to the patients enrolled in the study. So there is possible measurement error. They are not useful for investigating rare conditions, and they are reliable to survival bias. Then let's see a case control study. In a case control study, we consider a baseline characteristic of patients today, in the present. We divide the population according being a case or to be a control, meaning to have or not the disease. And then we check if in the past there was an exposure present or not. So it's substantially retrospective design. So also case control studies have merits and limitations. The merits are that they are suitable to investigate rare disease and outcomes, and they are not really expensive. They are not as cheap as cross-sectional studies, but they are not as expensive as cohort study or randomized clinical trial. The limitations are that they are not suitable for calculating frequency measures, they are not appropriate for rare exposures, and they are suitable to selection and recall biases. And it's important that there is this assumption that's very important for control studies. It's that not cases are representative of the source of population of cases. If this is not respected, we will have a selection bias. Tomorrow we will talk about this. Prospective cohort studies can have both a prospective or, cohort study can have both a prospective or a retrospective design. Let's see the prospective cohort study. If we start the study today, we measure the exposure and the confounder variables. Actually, we assess the characteristics of the study population. Then we discriminate among patients having or not a factor or an exposure, however we want to call it. And then we assess in the future if these patients developed or not the disease or had an outcome that can be, I don't know, all-cause mortality, cardiovascular events, or whatever. So this is a prospective cohort study, but a cohort study can be also retrospective, and we often see this in registry studies, for example. Even if we start the study today, for example, we consider baseline characteristics of patients 10 years ago, and according to those baseline characteristics, we divided the population in patients with and without the exposure, and then we check if from 10 years ago to today, these patients developed or not the disease, or there was an outcome. So also cohort study has merits and limitations. The merits are that there is temporary relationship between exposure and outcome, so it's not like a cross-sectional study that is performed regardless of follow-up. It allows to investigate several outcomes for each exposure, so we can have a study assessing several outcomes. For example, I don't know, in a study on a drug for myocardial infarction, we can at the same time assess the effect of the drug on stroke, I don't know, other cardiovascular outcomes, mortality or whatever. It is possible to perform matching, limiting, confounders rule. We will see this tomorrow. There are several ways we can use to limit the effect of confounders in this type of study, and for example, propensity score is one of these. They are easier and cheaper than a randomized clinical trial, and they are good to measure incidence of an outcome. The limitations are that they are more expensive than cross-sectional case control studies, and an outcome could take time to occur. Since often these are really long studies, developed in years, the definition of outcome can change over the time. And they don't have randomization, so there could be confounders driving our results in one or another direction. and there are also concerns about blinding and masking. So let's now see what the registry is. The registry is not a kind of study, but it's just a way to collect observational data, actually. They are defined as an organized system that uses observational study methods to collect uniform data, clinical or other, to evaluate specified outcomes for a population defined by a particular disease, condition, or exposure. So there is no restriction in time of type of collected data, and in principle there is no testing of research hypotheses. There are two types of registries. We have product registry. It means that they include all the subjects that receive the same drug or device, regardless of the disease or of the indication to use that drug or that device. And we can have disease registry that includes all patients with the same disease, actually, regardless of the drug or device exposure. So these are the keywords to summarize what a registry is, which ones are the most important characteristics of a registry study. It's not interventional. These emphasize that treatments are decided by healthcare provider and patients and not by a study protocol. So patients are just using a drug because according guidelines or the standard of care, they should receive that kind of treatment, and not because we want to. They don't receive that kind of drug because we want to perform the study. They are core study. That's an epidemiological term to define a group of study subjects with something in common who are followed up over time. And they are real world. This word is usually used to make a difference between observational studies and randomized clinical trial. In randomized clinical trial, the inclusion criteria are quite strict, and so sometimes we are worried that we cannot apply the results that we get from that kind of studies to the real world, in which there are really heterogeneous population. But registry study really enroll different types of patients, heterogeneous patients. So if we get a result from this kind of study, maybe it's more likely to can apply this observation to the real world. So these are the basic rules for data collection in a registry. The data are collected in a naturalistic manner. The registry is designed to fulfill specific purposes being defined before collecting the data. As we said, if it's a drug registry, for example, the specific purpose is to collect all the information about the efficacy, for example, of the drug. The registry captures information with specific and consistent a priori clinical definition. The data are collected in a uniform manner for every patient. The data collected reflect the clinical status of the patients. And registries includes the type of data that clinicians would use for diagnosis and management of patients. So let's talk about the data collection. Ideally, we should just collect the variables needed to achieve main objective. This is very important because if we plan to collect in a registry 1,000 variables, physicians or nurses or whoever is entering the data in the system will not do that because it's too much work. So we have to select the variable. We have to be realistic and precise. So as I was saying, if there are too many variables, there is also the risk to breach the non-interventional nature of the registry and to decrease the quality of data collection for core information. And it will be too much burden for physicians to include all this data in the registry, so there will be a lot of loss to follow up and a lot of missing data for specific variables. Registries are really important also in post-marketing surveillance in order to evaluate short and long-term effectiveness of a drug, to measure, monitor short, long-term safety and tolerability of a drug, for example, and to measure or improve quality of care. This is an example of a registry we have in Sweden. This is the Swedish Heart Failure Registry, RiksVikt. And I want to show you this just to understand how data are collected in a registry. So RxVict is a nationwide continuous health quality and research registry. It was founded in 2000, and actually there are around 60,000 patients enrolled in this registry. That's a national registry in Sweden, and it enrolls both outpatients and inpatients. The coverage is around 60%, and patients are enrolled both in cardiology and in internal medicine departments. As you can see, if we want to somehow make a difference between registry studies and randomized clinical trials, a registry study doesn't have so many inclusion criteria. Actually, the only inclusion criteria of the Swedish heart failure registry is to have clinician judged heart failure in inpatients or outpatients departments. And also, exclusion criteria are quite poor, quite few. The only exclusion criteria is the care in pediatric departments, so we don't enroll children, and if a patient decides not to be enrolled in the registry. The baseline data in our registry are collected at the time of hospital discharge or outpatient visit, and we actually collect around 100 variables. Data is entered by a local physician or nurse on paper CRF and then transferred to online CRF or directly entered to online CRF. Then the data are managed by the Uppsala Clinical Research Center. The outcomes don't come directly from the Swedish Heart Failure Registry, but from the population registry that is linked with the Swedish Heart Failure Registry. So the question is how it's possible to link different registries. Actually, in Sweden, every person living there has a personal number, and all the medical records refer to that personal number. So if actually a patient has atrial fibrillation, for example, and heart failure, the patient is at the same time enrolled in the Swedish Heart Failure Registry and the Registry of Atrial Fibrillation. And since these patients has this univoc personal number, it's possible to link the two registries and to collect variables from both registries. And that's the same way in which we collect outcomes, because we collect outcomes from the population registry, and actually we use the personal number in order to do that. There are no missing data for outcomes. Actually, we all know if a citizen die or not in Sweden. The only we have censoring just if the patient moves out of Sweden, actually. So these are the variables that are collected. Actually, we collect data about comorbidities from the National Board of Health and Welfare Registry. We collect even socioeconomic data from Statistics Sweden, that's another registry. So we have information about level of education, incoming, employment, utilization of social welfare, and so on. And we don't have missing data for these variables. We have a great coverage, and we can actually know all the drugs that these patients are taking, because every time that a person in Sweden goes to the pharmacy and takes a drug, this is registered, and using the personal number, we can know all the drugs that a patient was buying in pharmacies in Sweden. So this is just to show how our CRF is. It's 2 pages, around 100 variables. And as you can see, we have a lot of variables. Demographic variables as age, gender, socioeconomic as living arrangement, if a patient is an inpatient or a patient, if it was registered in cardiology or internal medicine, information about the follow-up, clinical history, if the patient is smoking or not, the duration of heart failure, clinical characteristics as the NIA class, the ejection fraction, clinical characteristics as blood pressure, heart rate, data on X-ray, actually, on kidney function, about potassium, hemoglobin, and NT-proBNP, all data about concomitant medications, as I was saying, that are actually both collected in the Swedish Heart Failure Registry and can also be got by the drug registry. And we have information about all the cardiovascular and not cardiovascular comorbidities. So as you can understand, this is a great tool both for research and for healthcare improving. So when we were talking about observational studies, as for randomized clinical trials and randomized trials, when we want to write a paper on observational studies, we have a flowchart that we can follow in order to be sure to write an appropriate paper, and this is called strobe statement, actually. So, just to summarize, registry and observational study have positive and negative aspects. The strengths are that they are representative of real world. They use large sample size, but there can be heterogeneity among investigative sites and patients. They have potential for improving standards of care. They are good for hypothesis generation. They are supportive data for label extension, and they are good for evidence-based medicine for outcome and reimbursement. The weakness actually are they are not randomized clinical trials, so there is space for confounders. There can be losses to follow up. There can be variability in data definition, outcome definition, and variables definition. There can be an under-reporting of side effects. They are subject to external validity. There could be biases, and people perceive observational studies as having a lower value as compared with randomized clinical trials. So comparing registries and randomized clinical trial, when we talk about registry, we talk about effectiveness, meaning the efficacy of a drug in the real world. Randomized clinical trial, we talk about efficacy, meaning like the efficacy of a drug in according strict inclusion and exclusion criteria of trial. Registries are observational, RCT are obviously randomized. We talk for registry about real world, and for randomized clinical trial, we have to consider the strict inclusion and exclusion criteria. Registries are hypothesis generating, while randomized clinical trial are hypothesis driven. Registry usually have a larger sample size as compared with randomized clinical trial. They are flexible, while randomized clinical trial are powered. And for registry, it's not required, GSCP, but it's optimal to have it. But for randomized clinical trial, it's required to have a GCP. So a few words about this. This is a new type of study. It's a registry-based randomized clinical trial. This is just an example. We are running in Sweden this trial called SPIRIT HFpEF in order to assess the efficacy of spironolactone in patients with heart failure and preserve the ejection fraction. I don't want to show you, the inclusion and exclusion criteria, but just how the study is planned. Actually, one major limiting factor in randomized clinical trial is how to enroll the patients. It's really difficult to enroll patients in randomized clinical trial. In registry-based randomized clinical trial, we use a registry in order to identify the patients that can be enrolled in the randomized clinical trial according inclusion and exclusion criteria. In this trial, for example, we are using Swedish Heart Failure Registry to check which patients are suitable for our trials. And after that, patients are called in hospitals in order to decide and check if they want to be enrolled or not. After that, they are randomized. They are randomized in using spironolactone or usual care. And another important thing is in randomized clinical trial, patients are really followed up quite often. There are really regular visits. In registry-based randomized clinical trial, we don't need these really regular visits because we can check clinical status of patients in the registry, actually, because if the patient is going to a physician, this is registered in our registry. So we don't need additional visits in order to run this kind of trial. And we don't need even this kind of follow-up in order to assess if an outcome happened or not, because we can check in the registry if the patient died or had a myocardial infarction, stroke, or whatever. So actually, it's really cheaper as compared with the randomized clinical trial. So if we compare these three types of study, randomized clinical trial have a really high quality study because they are randomized, but they are a huge list of limitations. They are very expensive. It's really difficult to have ethical approval for randomized clinical trial, and it's difficult to enroll patients. They are extremely expensive. The outcome assessment is manual, sometimes insufficient. And if we consider registry, there are a lot of positive aspects, as I was saying, but there is the big limitation that there is lack of randomized data, so they are really suitable to confounders. If we consider randomized, registry-based randomized clinical trial, they summarize the positive aspects of both. So they are cheaper. They are they need just a single ethical approval. The outcome assessment is automatic because we use the registry for that. It's easy to find patients to enroll because we use the registry for that. They are not selective, so we have real-world evidence because we use the registry for that. And so they can somehow summarize positive aspects of both registry and randomized clinical trial. So if you are interested to how research works with registry, this is a very nice guide that you can read, Registry for Evaluating Patients' Outcome. And from me, at the moment, is everything. If you have any question, really happy to answer. Thank you very much for your attention.